The living world is wonderful. We find living organisms in all types of habitats, like cold mountains, forests, oceans, freshwater lakes, deserts, and even hot springs. The diversity is amazing, isn't it? Life exists everywhere on Earth. What is life? What is the purpose of life? The second question is philosophical as a biology student. Let us learn what is life or living. What is living? You have already studied characteristics of living organisms. Growth, reproduction, metabolism, ability to sense environment and respond to it, and cellular organization are unique features of living organisms. Let us see how to define living. Growth is exhibited by all living organisms. Increase in body mass is considered as growth. Non-living objects also grow. Mountains and sand mounds show growth. However, the growth exhibited by non-living objects is by accumulation of material on the surface, while in living organisms, growth is from inside. Nevertheless, growth cannot be taken as a defining property of living organisms. Reproduction is a characteristic of living organisms. Organisms reproduce asexually or sexually and produce their own kind. However, there are living organisms which do not reproduce, example, mules, sterile worker bees, infertile human couples, etc. Hence, reproduction also cannot be the defining characteristic of living organisms. Metabolism is another characteristic of living organisms. There are various biochemical reactions which occur simultaneously inside all living organisms. The sum total of all the reactions occurring in the body of an organism is called metabolism. Non-living objects do not show metabolism, but metabolic reactions can be demonstrated in cell-free system, that is, in vitro. Hence, metabolism and cellular organization of the body is the defining feature of life form. Consciousness, technically, complicated feature of all living organisms is irritability. That is, their ability to sense their environment and respond to it. Both plants and animals respond to environmental factors like light, temperature, water, etc. All organisms are aware of their surroundings. Consciousness, therefore, is the defining property of living organisms. Thus, living organisms are self-replicating, evolving, and self-regulating interactive systems capable of responding to external stimuli. Biology is the story of life on Earth. It is an interesting life science. Diversity in living organisms. Enormous variety of living organisms exist on the earth. There are about 5 to 30 million species of plants and animals on earth. It is not possible for a human being to remember the characteristics of all these organisms without their systematic arrangement. Systematics. Systematics is the scientific study of similarities and differences among different kinds of organisms, and it also includes their identification, nomenclature, and classification. Taxonomy. It is the branch of biology which deals with the collection, identification, nomenclature, description, and classification of plants and animals. Objectives of systematics. First, to know various kinds of plants on the earth with their names, affinities, geographical distribution, habit, characteristics, and their economic importance. Second, to have a reference system for all organisms with which the scientists can work. Third, to demonstrate the manifold diversities of organisms and their phylogenetic, that is, evolutionary, relationship. Fourth, to ascertain nomenclature, that is, scientific name for every organism. The classification is found in day-to-day life. It makes the working very simple. Just imagine finding a book in the library where books are arranged in a haphazard manner. The library follows a well-defined system of arranging books. The stores and malls arrange their goods in a well-defined manner to facilitate the buyer to locate the right thing. Classification. It is the arrangement of organisms or groups of organisms in distinct categories in accordance with a particular and well-established plan. The classification is based on similarities and dissimilarities among the organisms. The term taxonomy was coined by A.P. de Candolle in 1813. So what exactly was the need of classification? Well, there is a large number of living organisms found on Earth. All these organisms show variations in their shape, size, structure, habit, habitat, nutrition, etc. It is very difficult to remember the characteristics of all the organisms without their proper arrangement. The classification helps to explain unity in diversity of the organisms. It gives specific and scientific names to the organisms which are accepted universally. It reveals the relationships among various groups of organisms. The classification places an organism amongst those which have common characteristics. The three domains of life. The three-domain system was developed by Carl Woese in 1990. It is a system for classifying living organisms. From the late 1960s, organisms are being classified according to a five-kingdom system. Classification systems change as scientists learn more about organisms. Genetic sequencing has given researchers a whole new way of analyzing relationships between organisms. The three-domain system is primarily based on differences in ribosomal RNA structure of different groups of organisms. Ribosomal RNA is a molecular building block for ribosome. According to this system, organisms are classified into 3 domains and 6 kingdoms. The 3 domains are archaea, bacteria, and eukarya. The domains archaea and bacteria include prokaryotic organisms. These domains include only 1 kingdom each, that is, archaea bacteria, which is ancient bacteria, and eubacteria, that is, true bacteria, respectively. The domain eukarya includes all eukaryotes. The 4 kingdoms under this domain are kingdom, Protista, fungi, Plantae, and Animalia. Taxonomic hierarchy. The Greek word hierarchs means rank. The main aim of taxonomic study is to assign each organism an appropriate place in a systematic framework of classification. The manner of scientific grouping of different taxonomic categories in a descending order on the basis of their ranks or positions in classification is called taxonomic hierarchy. In the system of classification, the terms like taxa and categories are often used. 1. Taxon. Taxon is a group of living organisms. Its plural is taxa, which is used to represent a concrete unit of classification. The unit of taxon may be large or small. The term taxon was coined by H. J. Lamb in 1948 for plants. 2. Category. A category is a rank or level in the hierarchical classification of organisms. Various taxa are assigned definite categories according to their taxonomic status. In the hierarchy of categories, kingdom is the highest and species is the lowest category. Example: method of classification of China rose and cobra. First, let's classify China rose. Kingdom Plantae. Its division or phylum is Angiospermae. Class Dicotyledone. Subclass Polypetalae. Series Thalamiflorae. Order Malveolus. Family Malvaceae. Genus Hibiscus. And species Rosa sinensis. Next, let's classify cobra. Kingdom Animalia. Division or phylum Chordata. Class Reptilia. Subclass Diapsida. Series Order Squamata. Family Elapidae. Genus Naja. Species Naja. Units of classification: kingdom, division or phylum, class, subclass, series, order, family, genus, and species. In the hierarchy of categories, kingdom is the highest and species is the lowest. Hierarchy of taxonomic categories. Species. Species is the smallest and basic unit in the system of classification. All the individual members belonging to a particular species show all similar characters and can breed among themselves to produce similar type of organisms. Example, all the China rose plants are grouped under a species Rosa sinensis. Similarly, all the potato plants are grouped under a species Tuberosum. Concept of species. There are different concepts of species as morphological concept given by Carlos Linnaeus, biological concept given by Charles Darwin, and modern concept given by Ernst Mayr. According to Linnaeus, a group of organisms which are similar in their morphological characters is called morphospecies. According to Darwin, a group of organisms that can interbreed under natural conditions is called biospecies or true species. The modern concept, according to Ernst Mayr, a group of actually or potentially interbreeding natural population of closely resembling organisms is called species. Genus. Its plural is genera. It is a group of closely related species which resemble one another in certain characters. Example, Rosa sinensis, Esculenthus, Cannabinus, etc. are different species under the genus Hibiscus. A genus may be either monotypic, that is having single species, or polytypic genus, that is having many species. Family. A family represents a group of closely related genera. The genera like Hibiscus, Malva, Cedar, Gossypium, Abutilon, etc. belong to the family Malvaceae. Order. It is a group of closely related families which resemble in major characters. For example, families Malvaceae, Tiliaceae, Sterraculiaceae, etc. belong to the order Malvalus. They show axile placentation in ovary. Subclass. It is an assemblage of different orders having some similarities. For example, the orders Malvalus, Ranales, Varietales, etc. have free petals and hence grouped under the subclass Polypetalae. Class. It is a group of related subclasses. For example, the class Dicotyledone includes subclasses as Polypetalae and Gamopetalae. Division. The division is a category composed of related classes. For example, division Angiospermi includes two classes, dicotyledony and monocotyledony. In animal classification, instead of division, the category phylum is used. Subkingdom. Different divisions having some similarities form subkingdom. For example, the divisions Angiospermy and Gymnospermy will form the subkingdom for narrowgams or Spermatophyta. That is, all seed-producing plants. Kingdom. It is the highest taxonomic category composed of different subkingdoms. For example, subkingdom for narrowgams and cryptogams form the plant kingdom or Plantae, which includes all the plants, while all animals are included in kingdom Animalia. Nomenclature. Nomen means name. Calare means to call. Any object that becomes known to human intelligence is given a name. Just as all known objects are given names, plants and animals also are identified by their names. The art of naming an object is in fact a science called nomenclature. The nomenclature has two main purposes. First, as an aid to communicate, and second, to indicate relationship. Vernacular names. These are the names which are given to organisms in a particular region and language by local people. All plants and animals are known by different common names in different parts of the world. Sometimes, within the same country, people of different states and regions use different common names for a single organism. The local or vernacular names are short, familiar, and easy to follow. However, these names do not communicate the necessary information and do not indicate proper relationship and are not universal. For example, in India, Epomia batatas is recognized by various names like sweet potato in English, shakarkand in Hindi, meetha aloo in Assamese and Bengali, kandamul in Telugu, ratalu in Marathi, and janasu in Kannada, etc. It means the local names are different and so confusing. A single vernacular name is used for several species. For example, the name lily is used to describe many bulbous flowering plants like water lily, spider lily, etc. Vernacular names may be misleading. For example, starfish, jellyfish, silverfish are not fishes at all. Thus, vernacular names create confusion. Scientific names. These names are based on some criteria and principles which are accepted all over the world. These names were earlier polynomial. Example, Sida acuta, a member of family Malvaceae, was named as Chrysophyllum, Polis, Ovalis supreme, Glaberus, Parallel, Striatus subtus, Tomento sonitidis. Such long names are difficult to remember, are they not? Hence, to make it easier and meaningful, binomial system of nomenclature was introduced. Binomial Nomenclature. The credit of giving binomial system of nomenclature goes to the Swedish naturalist Carolus Linnaeus. He introduced the system in his book Species Plantarum, published in 1753. A system of nomenclature of plants and animals in which the scientific name consists of two words or parts or epithets is called binomial nomenclature. According to this system, the scientific name of sunflower is Helianthus annuus. In the above name, the first word, Helianthus, indicates name of the genus, that is the generic name, and the second word, annuus, denotes name of the species, that is specific name to which the plant belongs. The binomial nomenclature system follows certain rules, such as the name of the organism is composed of two Latin or Latinized or Greek words. The generic name is a simple noun which should come first and should begin with a capital letter, while specific name is the descriptive adjective which should come later and begin with a small letter. These names are based on some special characters of the organism. These specific names must be underlined separately if handwritten and must be printed in italics. The generic as well as specific name does not generally have less than 3 letters and more than 13 letters. Usually the name of the author who names a plant or animal is also written in full or in abbreviated form after the scientific name. Example, Mangifera indica L, where L stands for Linnaeus. Mention of author's name after the species name is called citation. To avoid the confusion, no two generic names in any kingdom can be same. However, the species name can be repeated when the genera are different. For example, Mangifera indica, that is mango, and Azadirachta indica, that is neem. When a single species is described under different names by different authors, then these names are called synonyms. In such cases, the name under which the species is first described is considered to be valid. The binomial nomenclature has certain advantages. These names are simple, meaningful, precise, and standard as they are accepted universally. Due to this system, confusion and uncertainty created by local or vernacular names is avoided. The binomials are easy to understand and remember, indicate phylogeny, that is, evolutionary history of organisms, and help to understand relationship, better organisms, and group organisms. 5-kingdom system of classification. The 2-kingdom system of classification of organisms was introduced by Carl Linnaeus. The system recognized 2 kingdoms of organisms as Plantae and Animalia. However, this system was found inadequate for classification of some organisms like bacteria, fungi, euglena, etc. Haeckel suggested the 3rd kingdom, Protista, to include these organisms. The system of classification proposed by R. H. Whitaker in 1969 is a 5-kingdom system. This system shows the phylogenetic relationship between organisms. The 5 kingdoms are 1. Kingdom Monera, 2. Kingdom Protista, 3. Kingdom Fungi, 4. Kingdom Plantae, 5. Kingdom Animalia. The criteria used by Whitaker in system include: first, cell organization, whether the organism is prokaryotic or eukaryotic. Second, body organization, whether the organism is unicellular or multicellular. Third, mode of nutrition, whether the organism is autotrophic or heterotrophic. Fourth, lifestyle, whether the organism is producer, consumer, or decomposer. Kingdom Monera. The organisms included in this kingdom are microscopic and prokaryotic, having a primitive type of nucleus. Pro means first formed, karyon means nucleus. Prokaryotic nucleus is also called nucleoid and is not well organized. It is without nuclear envelope, nucleolus, nucleoplasm, histone proteins, and true chromosomes. That is, only DNA is present. The cell wall is rigid and composed of peptidoglycan. These organisms are unicellular, as in eubacteria, or filamentous, as in cyanobacteria. The cells are without membrane-bound cell organelles like chloroplasts, mitochondria, etc. These organisms are either motile, that is, the locomotion is due to gliding movement of flagella, cilia is absent, or non-motile. The organisms exhibit different modes of nutrition like autotrophic, heterotrophic, parasitic, or saprophytic. The autotrophs are either photoautotrophs, that is, cyanobacteria, or chemoautotrophs, example, Thiobacillus. The mode of reproduction in Monera is either asexual or with the help of fission or budding. Sexual reproduction, mitosis, and meiosis are absent. Examples: Archaebacteria, example, Methanobacillus, Thiobacillus, etc. Eubacteria, example, Rhizobium, Clostridium, etc. Cyanobacteria, example, Nostoc, Anabina, etc. Actinomycetes, example, Streptomyces, Mycobacterium, etc. Kingdom Protista or Protoctista. These organisms are unicellular, eukaryotic, and mostly aquatic. However, some are terrestrial. They are either motile or non-motile. The locomotory organs are cilia, flagella, pseudopodia, etc. The protists are with or without cell wall. If cell wall is present, it is composed of cellulose. They have membrane-bound cell organelles such as mitochondria, chloroplasts, ER, Golgi complex, etc. The nucleus is well organized, that is, with nuclear envelope, nucleoplasm, nucleolus, and helical DNA with histone proteins forming chromosomes. They show either photosynthetic, holozoic, saprophytic, or parasitic mode of nutrition. The reserve food material is in the form of glycogen, or starch, and fat. The reproduction is by asexual or sexual method. Asexual reproduction occurs by mitotic or amitotic cell division, while sexual reproduction is by fusion of male and female gametes forming zygotes. The zygote directly develops into young individual without formation of embryo. Protists may further be distinguished into A. Plant-like protists. They are autotrophic, that is photosynthetic, have cellulosic cell wall, and store reserve food as starch. Example, dinoflagellates, diatoms, and desmids, that is golden algae. They are together grouped as chrysophytes. The cell wall of diatoms is composed of two overlapping halves resembling a soap case. The remains of the cell walls is called diatomaceous earth, which is used in chromatography, filtration, and polishing. They are chief producers of aquatic environment. B. Animal-like protists. These are heterotrophic, ingest food, lack cell wall, and store reserve food as glycogen. Example, amoeba, paramecium. C. Fungi like protists. They are saprophytic and lack cell wall. Example, slime molds. D. Euglenoids. These are primarily photosynthetic aquatic organisms. However, they behave as heterotrophs in the absence of light. Example, Euglena. Thus, kingdom Protista is a link between prokaryotic kingdom Manura and complex eukaryotic kingdom Plantae, fungi, and Animalia. Kingdom Fungi. It is a unique kingdom of heterotrophic organisms showing extracellular digestion. The organisms are unicellular, like yeast, or multicellular and filamentous, like penicillium. Unicellular organisms have a protoplast with many nuclei. Example, rhizopus, or with a single nucleus. Example, yeast. Filamentous organisms consist of a body called mycelium, in which a number of thread-like structures called hyphae are present. The hyphae may be with septa, that is septate, or without septa, that is, aseptate. They may be uni or multinucleated. The non-septate multinucleated hyphae are called phenocytic hyphae. The cell wall in fungi is composed of chitin or fungal cellulose. Fungi contains well-organized membrane-bound cell organelles except the chloroplasts. The fungi exhibit heterotrophic mode of nutrition. Most of the members are saprophytes and absorb food which is decomposed, that is digested outside. Thus, absorption is the mode of nutrition. Some are parasites or predators and some are symbiotic. Reproduction is vegetative, asexual, or sexual. The fungi are further classified as follows. Phycomycetes. These are commonly called algal fungi and consist of phenocytic hyphae. They commonly grow in moist and damp habitats on decaying organic matter. They reproduce by endogenous spores produced inside sporangia. Examples: Muca, Rhizopus, etc. Abugo is an example of parasitic Phycomycetes. Ascomycetes. They are mostly multicellular and are commonly called sac fungi. When multicellular, their thallus or somatic body is composed of branched septate hyphae. They produce endogenous sexual spores called ascospores in sac-like structures called asci. Some produce fruiting bodies, that is ascocarps, which enclose asci. The asexual and exogenously produced spores are called conidia, which are at the tips of special hyphae called conidiophores. Examples: Aspergillus, Penicillium, Neurospora, etc. Yeast is an example of unicellular Ascomycetes. Basidiomycetes. These fungi, commonly called club fungi, include mushrooms, bracket fungi, rusts, smuts, etc. They have branched septate hyphae. Asexual reproduction is absent, but vegetative reproduction takes place by fragmentation. Sex organs are lacking. The fusion of two vegetative cells or somatic cells, that is somatogamy, produces a dikaryotic mycelium which gives rise to basidia or a fruiting body called basidiocarp. Basidiocarp contains many basidia which produce sexual spores called basidiospores exogenously. Examples: Agaricus, Astilago, Puccinia, etc. Deuteromycetes. It is a temporary group of fungi which are known to reproduce only asexually. They are commonly called imperfect fungi. Whenever their sexual reproduction is discovered, they are transferred to the respective groups. They are mostly decomposers, while a few are parasitic. Examples: Alternaria, Coletotrichum, Trichophyton, etc. Kingdom Plantae. The kingdom Plantae includes eukaryotic and autotrophic organisms like algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. The plants show sedentary nature. The cell wall of plant cell is composed of true cellulose. The members possess photosynthetic pigments. The members of Plantae are the main producers and provide food directly or indirectly to all living organisms. Reserve food material is in the form of starch. Vascular tissues are absent in the lower plants, that is algae and bryophytes, but present in higher plants, that is pteridophytes, gymnosperms and angiosperms. In higher plants, cuticle, stomata, etc. are present to check the rate of transpiration. The plants reproduce by vegetative, asexual, or sexual methods. Vegetative reproduction occurs with the help of vegetative parts, asexual reproduction with the help of spores, while sexual reproduction takes place with the help of gametes. Examples: algae, example, Spirogyra, Chara, etc. Bryophytes, example, Riccia, Funaria, etc. Pteridophytes, example, Lycopodium, Nephrolopis. Gymnosperms, example, Cycas, Pinus, etc. Angiosperms, example, sunflower, jowar, etc. Kingdom Animalia. The animals are multicellular and eukaryotic and may be aquatic, terrestrial, amphibious, and aerial. Majority of the animals are motile. However, a few, like sponges, are sedentary. The animal cells do not possess cell wall, plastids, and central vacuole. The animals possess sense organs, nervous system, and they respond to stimuli by exhibiting certain behavior. They are heterotrophic, mostly holozoic, and some are parasitic. They are consumers of various levels: primary, secondary, tertiary, etc. Examples are fishes, frog, cats, birds, etc. The five-kingdom system of classification has some advantages, as prokaryotes are separated from other organisms, that is, eukaryotes, and get the status of kingdom. The fungi are separated from Plantae on the basis of their saprophytic mode of nutrition, that is, absorption. Thus, all groups of organisms get proper taxonomic status, example, rank of kingdom, Monera, and fungi. Euglena, an organism showing photosynthetic mode of nutrition in presence of light, and holozoic mode, that is ingestion of food in absence of light, has been placed in kingdom Protista as it is unicellular. Thus, organisms with doubtful placement like Euglena get proper place in classification. During redistribution of the organisms among additional kingdoms, the original subdivisions of old two-kingdom system are not disturbed. However, there are some drawbacks in five-kingdom system. Viruses do not find any place in five-kingdom system of classification. Kingdom Protista includes photosynthetic and non-photosynthetic organisms as well as those with or without cell wall, and it is difficult to imagine them together. Lichens. The lichens are slow-growing, long-living organisms occurring in wide variety of places ranging from seashore to Arctic and Antarctic regions of the earth. Theophrastus in 300 BC was the first to use the word lichen. Lichens are formed by close association of two different partners, that is organisms, one of which is an algal component and the other is fungal component. The algal component is called phycobiont or photobiont, and it mostly belongs to Chlorophyceae, that is green algae, or cyanobacteria, that is blue-green algae. The fungal component is called mycobiont, and it mostly belongs to Ascomycetes and rarely Basidiomycetes or Deuteromycetes. The lichens are distributed in wide variety of habitats. They grow on tree trunks, decaying logs, and on soil too. They are found in extreme climatic conditions such as on dry exposed rocks and also at snow-covered Arctic and Antarctic poles. They are sensitive to air pollution and generally do not grow near the cities. They are thalloid and the thallus is greenish or bluish-green in color. Some lichens have additional pigments such as yellow, orange, brown, and black. On the basis of fungal components, Alexopoulos and Mims in 1979 divided the lichens into three categories as A. Ascolichens. In this category, the fungal partner belongs to Ascomycetes. B. Basidiolichens. Here, the fungal partner belongs to Basidiomycetes. C. Deuterolichens. In this category, the fungal partner belongs to Deuteromycetes. These are sterile lichens producing no spores. On the basis of external form, that is thallus organization, the lichens are of three types as A. Crustose lichens. These are thin and flat lichens occurring as crust on the bark of rock. Example, Graphis, Lecanora, Hematoma, etc. B. Foliose lichens. These are lobed and dorsi-ventrally flattened, leafy lichens. They are attached to substratum by hairy rhizoid-like structures called rhizemes. Example, Parmelia, Collema, Peltigera. C. Fruticose lichens. These lichens are commonly called shrubby lichens. They have cylindrical, branched, and erect or pendulous thallus, which gives them a shrub-like appearance. They remain attached to the substratum by basal mucilaginous disc. Example, Cladonia, Usnea, Alectoria. The lichens generally reproduce by vegetative, asexual, and sexual methods. Economic importance of lichens. 1. Lichens as food. Several lichens are used as food. The lichens contain a substance, lichinin, which is similar to carbohydrate. Example, Lecanora esculenta in Israel and Umbilicaria esculenta in Japan. Parmelia is used in curry powder as well as in making chocolates and pastries. 2. Lichens as fodder. Lichens form a favorite food for reindeers and cattle. Species of Cladonia, Citraria, Evernia, Parmelia are used as fodder. 3. Medicinal uses. Usnic acid obtained from the Usnea and Cladonia species is used as an antibiotic against gram-positive bacteria. Laboria, Citraria species are used in respiratory diseases like TB. Peltigera is useful in hydrophobia. Parmelia is useful in epilepsy, and Usnea species in urinary disease. Some lichens possess anti-carcinogenic property. 4. Industrial uses of lichens. In Sweden and Russia, lichens are used for production of alcohol. Lichens are used in the process of tanning and dyeing. In the litmus paper preparation, which is used as acid-base indicator extract of species of Rossella and Lesalia are used. Orthene, a biological stain, is obtained from Orcolechia and Androgyna and O. Tartaria. In perfumery, species of Evernia and Ranolina are the sources of essential oils which are used in preparation of soaps and other cosmetics. Ecological importance of lichens. The lichens are the pioneers of vegetation on rocks. Lichens are the first plants to settle on barren rocks. Lichens bring about weathering of rocks by releasing carbonic and oxalic acids. Weathering of rocks leads to formation of soil and is called pedogenesis. After lichens, some bryophytes and then other higher plants can grow on such soil. Viruses and viroids. Viruses. Viruses are acellular, highly infectious microorganisms which are considered to be on the threshold of life. Adolf Meyer in 1886 had demonstrated that mosaic disease of tobacco is infectious. When the juice from tobacco plants infected with the tobacco mosaic virus, that is TMV, was injected into healthy plants, the plants showed symptoms of mosaic disease. D.J. Ivanovsky in 1892 confirmed the observation of Mayer. Ivanovsky prepared an extract of infected tobacco plant and showed that this extract could infect healthy plants even after it was passed through filter that retains bacteria. M.W. Beechering in 1898 called it as virus venom or poisonous fluid and referred it as contagium vivum fluidum, meaning contagious living fluid. The term virus was first used by Louis Pasteur. Today, our knowledge of viruses is due to efforts of scientists like Frederick William Twort, Felix de Herille, Skelsinger, W.M. Stanley, Hershey, and Chase, etc. The detailed structure of viruses was studied only after the invention of electron microscope. Viruses are acellular, ultramicroscopic, disease-causing, that is, infectious entities. Their size ranges from 10 nanometer to 2000 nanometer. Some of the larger viruses are, however, as large as some of the smaller bacteria. Viruses do not grow, do not eat food, or respire. Viruses are obligate parasites and can multiply only within the living host cells. They have capacity to transmit the disease from diseased to healthy organisms. Viruses show very simple structure, that is a nucleic acid core surrounded by a protein coat, that is capsid, to form the nucleocapsid. They possess their own genetic material in the form of either DNA or RNA, but never both. They can be crystallized. Stanley, in 1935, obtained viruses in crystalline form from tobacco leaves infected by TMV. They can be stored for long period. Outside the body of host, they do not show any activity. They are like non-living. They do not have their own metabolic machinery. They utilize the ribosomes and enzymes of the host cell for synthesis of protein during reproduction. Most of the viruses are highly resistant to germicides and extremes of physical conditions. Viruses exhibit mutations. They cannot be cultured on artificial nutrient medium. Viruses are host-specific. Thus, viruses show characteristics of living as well as non-living. Therefore, they are said to be on the threshold of life. Viroids. Viroids were discovered and named by Theodore Diener in 1971. These are very small, circular, single-stranded RNAs which are not complexed with any protein, that is without protein coat. This RNA is either covalently closed circular RNA or single-stranded linear RNA. These are mainly plant pathogens. The viroids do not show dormant phase. The first viroid discovered was the potato spindle tuber viroid, that is PSTV, which causes a disease in potato. PSTV is composed of 359 nucleotides and thus has 10 times less genetic material than the smallest known virus. The naked RNA molecule can replicate autonomously in plant cells. It can also disperse into the environment and produce diseases. At least 11 other plant diseases have been linked to viroids. These diseases include Citrus exocortis, Chrysanthemum stunt, Cucumber pale fruit, etc.